All right, it is 12:30, so let us get started. So reminder that your test is Thursday, almost said tomorrow. And the stuff that we cover today, any stuff that has to do with lipids will not be on your test. So if you've looked at the old test 2 from last year, that was able to give in person before the university went all online, you can see that I have questions on there about lipids and beta oxidation, the breakdown of lipids. None of that will appear on your test 2. So just remember this end of test 2 material in the slide. Everything before that, oxidative phosphorylation, citric acid cycle, gluconeogenesis, pyruvate dehydrogenase, that's all fair game. Nothing to do with lipid breakdown or transportation. The test itself, I'm just about finished writing it. My aim is to have roughly 40 questions, like 32 of them will be multiple choice. It's roughly the ratio I'm looking for. You'll have an hour and 15 minutes, so that should be plenty of time to answer all of those questions. Let's see. Is there anything else I want to cover about the test? So what's on today's reading guide won't be on the new test. Correct. Anything that we talk about today won't be on test 2. It will be on test 3. Yeah, breaking down of lipids, all that stuff is on test 3. Yeah, I don't think I have anything else to say really about the test. You know, it's going to be ProctorU, all the normal stuff. You can have scratch paper, all that. You can have a calculator. Yeah, if you have questions about the test, feel free to put them into chat if you want to know anything else about the test. And we'll probably have a little bit of time afterwards, after like we get through the material today, to maybe go over a few specific questions. But does anyone have any general questions they are thinking of right now that they want to get out of the way? Well, if you do think of something, feel free to let me know. So just a little review of what we talked about on Thursday. We were talking about how to digest triacylglycerol. So we're getting into fat metabolism now, starting with the breakdown of fats. So we looked at, you know, how when eating fat, how do you transfer it from your small intestine into your cells? How are the various ways that you can move it through the bloodstream, what each of those carriers are. And we also talked about LDL, uptaking that into your cell and how that is broken down, right? So this is the slide we ended up on, talking about the digestion of LDL. So let's actually talk about the LDL molecule itself a little bit more in depth. So it's made out of more or less a few components. Let me get my actual pen out here. So you have a protein component, which we call the apolipoprotein. You have your cholesterol, and then you have some phospholipids. And there's a little bit of fatty acid in there still, but this is mainly a cholesterol carrier. Now, we know that there are roughly 9 different types of proteins that can associate with our lipoproteins, or associate with our lipid carriers. And depending on, you know, what lipoprotein you're looking at, they're going to be in different amounts. So we're going to focus on ApoB100. This is the lipoprotein that associates with our low density lipoprotein, our LDL. This molecule, as you would expect, is extremely hydrophobic, and it's shown over here of what the amino acid makeup of ApoB100 is. So it has nonpolar residues. That's going to interact with the actual lipid and cholesterol portion of your molecule. And then it has a polar portion, which will interact with the water. So this protein, as shown in this artistic representation, is just kind of wrapping itself around this molecule, our fat cholesterol molecule. And it's wrapping around and forming these helices where, you know, the nonpolar part of the helices are pointing towards our cholesterol. The polar part are facing towards the water. And roughly every LDL will have one ApoB100, covers half of this molecule. So it's actually covering more than what is being shown in this artistic representation. So these lipoproteins that we talked about, as the name suggests, has a protein in it. So they're just not fat globules or cholesterol globules moving through your blood, but they also have this protein that helps them be soluble and helps them move across into different cell types, helps them to be recognized. And based on what lipoprotein you're talking about, you'll have a different apolipoprotein associated with it. So just, I know that terminology gets really confusing, but just when you talk about an apolipoprotein or apoprotein, that's just the protein part, and the lipoprotein is the whole complex. The lipoprotein would be the lipids plus the protein. So that is how our LDL is looking when it's moving through the bloodstream. And if we go back to our previous slide that we ended up talking about on Thursday, you can see when we break down our LDL, remember that's what we're doing here. So ApoB is the protein we just talked about. That is what is being recognized in our clathrin-coated pit. So that's how cells know, this molecule is LDL, because only LDLs have this ApoB protein. So our receptors are binding to the protein. And then when our LDL is broken down, you can see here we are spitting out the amino acids of that protein. So we break down the amino acids, and any fatty acids that are still with our LDL, we break those down in our lysosome and spit those out into the cell so they can be used for other purposes, while the cholesterol continues on to the ER, like we talked about on Thursday. So that's how we can relate the structure of LDL to what is going on in this image. So any questions to begin with LDL, apolipoproteins, or anything along those lines? All right. So let's just take a little little side venture here and talk about why LDL is called the bad cholesterol. It makes sense why HDL is called the good cholesterol, because HDL, high density lipoprotein, this is the molecule that takes cholesterol from your cells and delivers it to the liver, while LDL is the molecule that takes it from the liver and can deliver it to your cells. So what can happen to cause blockages and cholesterol buildup inside your body? Well, sometimes when LDL goes to deliver cholesterol to cells, it'll get trapped in the proteoglycan layer. So remember, we briefly talked about this in biochemistry 1, but it's just basically on the outside of some cells, you have this sugar and protein like matrix. And LDL can get trapped there. And then it starts to get oxidized, starts doing some redox reactions. This is not what's supposed to happen to LDL. So to try and clean this up, what happens is that white blood cells are recruited to this. White blood cells will come over and try to clean up this LDL that has been trapped in the proteoglycan layer. But what can happen is that these white blood cells can actually eat too much of the fatty acids. It can eat too much of the cholesterol. And white blood cells, they're not designed to really metabolize all that cholesterol, all that fatty acid. So what happens is they become what are called foam cells. And they're called foam cells because when you look at them, since all that fat and cholesterol, it makes them look really foamy. And sometimes what happens is that in your blood vessel wall, right, your white blood cells will go inside there, eat all the fat that's leaked in there, all the cholesterol, and then they will die and kind of just stick around on the vessel wall there. Well, if they die and the reason that they die is that they eat too much fat and cholesterol that just overwhelms the cell and the white blood cell dies. So what happens is that other white blood cells will come and try to eat all the fat and cholesterol that's still being released, and it's still stuck there because the white blood cell didn't actually take care of it because it died. And then those white blood cells become foam cells and they die. And that just continues on where, okay, white blood cell comes and tries to eat all that cholesterol. There's too much, it dies until more and more cells just pack up on there, and cholesterol is also in there, and you get this nice hard plaque. And that is one of the reasons you can have stroke or heart attack, or that in general is what's happening with your arteries and artery diseases. And so that's why, if you have high levels of LDL, what that is telling us is that you have high levels of cholesterol floating around in your bloodstream, mainly due to your diet. If you eat a lot of cholesterol in your diet, which will cause this plaque to eventually build up. Luckily, this is reversible if you help to change your diet. As we saw, the HDL can start taking away some of this cholesterol and to start reduce some of this plaque. But it's best to try and not to get this plaque started. So that in a nutshell is the biological reason for LDL and HDL being called the way they are. Any questions about that idea? All right, if not, we can move on. And so here's one of our critical thinking questions. So let's say that you decide to become a doctor and you're working in a hospital, and then a child comes in, really bad stomach pains after eating hamburgers, fries, and ice cream, the typical American lunch. And the blood of the child, when you look at it, looks like cream tomato soup. So this is weird. So you do an analysis in the blood and you see, oh, there are a lot of chylomicrons in the blood. What is happening then? And how would you prescribe to fix this problem for this patient? So I'll give you a few minutes to think about this, and like take a minute or two, and then I'll just come back with the answer. So the hints are in this image showing you what's happening. Think about why something like this would happen. And I'll be back in a minute. All right, so hopefully you had a little bit of time to just start to percolate ideas on what might be happening to this patient. So if we review the information, if we have a lot of chylomicrons in our blood still hours after eating a very fatty meal, what is the most likely explanation is that this person has a defective lipoprotein lipase. Remember, lipoprotein lipase is the enzyme that will break down chylomicrons into fatty acids and cholesterol. So this isn't happening to this person. So the treatment for such a condition is that you would have to restrict their diet. You just have to say you can't really eat fatty foods anymore because you can't digest them properly. You can't break them down in your bloodstream. And as long as you have a manageable diet, you should be able to prevent something like this from happening again. So this was just a question to get you back to reviewing how this whole system works again. But with that, that should be the last PowerPoint slide we have for what we started on Thursday. So let me just switch over to the slides I've prepared for today. All right. And what we're going to be talking about today is just the biochemical way we break down fatty acids, which is called oxidation or beta oxidation. So if you need energy, one of the ways to get energy is through fatty acids. That's your main energy storage, actually. And so let's say you're running low on energy. Well, lipase, and it's hormone sensitive. What that means is that, once you have the hormones released for like, I'm hungry, lipase can go to your adipocytes or your fat cells and release triacylglycerols so they can start to be used for energy. Now, these fatty acids, the first thing you need to do is break them out of glycerol. So you just break off the tails. And once that is done, these fatty acids can bind to serum albumin in the blood. And they have to do this because fatty acids are extremely nonpolar, so they can't travel through the blood by themselves. So this serum albumin is like a little protein boat that they like climb aboard on, and that can deliver them to the tissues that need the energy. Mostly your muscles are going to be getting a lot of that fatty acid. And like I said, the breakdown of fatty acids, we call this beta oxidation, and we'll look at why that's called beta oxidation today. But the very first step in doing this is that we have to prime our fatty acid using ATP to form what's called fatty acyl coenzyme A. So even before breaking down any fatty acids, we have to burn an ATP. And we use 2 phosphates to do this process to make AMP. But the idea here is we attach a fatty acid to an AMP molecule. That's what this acyl adenylate mixed anhydride is. It's just AMP plus fatty acid. And then our friend coenzyme A, which we saw in the citric acid cycle. So if you remember, which you're going to be tested on Thursday, one of the first steps, or the first step of the citric acid cycle that we talked about, is that we take pyruvate and make it into Acyl-CoA. Acyl-CoA is a very high energy molecule because of this sulfur bond. And so we're going to make fatty acid Acyl-CoA here, where this R is just our fatty acid chain. So that's going to be our first step in this process, making our fatty acid coenzyme A. And this is done by Acyl-CoA synthetase. Synthetase is usually something that makes, it's an enzyme that makes something. Here we're making acyl coenzyme A. So that's the first step in our fatty acid oxidation process. Now, we have our little carrier of fatty acid, the serum albumin, and we can deliver that to the cell, no problem. However, the breakdown of this fatty acid, that happens in the mitochondria. And we had a similar problem when we were talking about gluconeogenesis and the citric acid cycle, that the mitochondria is selective and doesn't have a lot of carriers. And the problem here is there is no carrier for Acyl-CoA. We cannot take Acyl-CoA from the cytosol, move it across the inner mitochondria membrane to the matrix. We don't have that protein, that transporter. So we need to attach it to something else to carry it. And that's why we attach it to carnitine. If you ever gone to the, I forgot what those stores are called, like GMC or something like that. Anyways, if you go to like the vitamin, there we go. I don't know why I was not thinking of that word. Go to the vitamin aisle in a grocery store, you'll see that you can buy this stuff. And they say, you know, this will boost your workouts. Well, let's look at the biochemical basis for why they might say that, even though, you know, probably just eating this stuff by itself won't do much to you. So what carnitine is, it's just a molecule that looks like this. And it will bind onto your fatty acid. So you're going to kick off coenzyme A, and you're going to transfer this fatty acid to make an acyl carnitine. And that's what's being shown here. Once you have a carnitine, your carnitine carrier protein has no problem moving it across the inner mitochondrial membrane. Even when it's attached to our fatty acid here, we can still move this complex from the cytosol to the matrix of the mitochondria. However, like I said on the previous slide, I can only start beta oxidation if I have an acyl coenzyme A fatty acid. So I just have to reverse the process, what I just did. Instead of taking off a coenzyme A inside the matrix, I just have to reattach my coenzyme A. And so the enzymes that do this are carnitine palmitoyl transferase 1 and 2. 1 is in the cytoplasm, removes our coenzyme A. 2 is in the matrix, reattaches our coenzyme A. And carnitine carrier protein is just moving carnitine. You can do it with or without the fatty acid. All right, so that's how we're going to get our fatty acids from the cell into the matrix so we can start our beta oxidation process. So up to this point, any questions either of you might have? As we are down to 2 people for the live lecture, although I guess, since there's a test on Thursday, it's not that surprising. All right. So first kind of like thinking question here. So you can get a condition where carnitine palmitoyltransferase 2 does not work. So you can get a condition where you can never reattach coenzyme A to your fatty acid after bringing it across with carnitine. So why would these individuals have naturally weak muscles? And why are these symptoms more severe during a fasting condition? Based on the information we just talked about, take a minute, think about that, and then I'll just come back with the answer after a minute. So use this time, just review, make sure you understand all the information that I went over, and I'll go over the answer here in a minute. All right, so in this condition, carnitine palmitoyltransferase 2 does not work. Yep, so that's correct. So this fatty acid, we can break that down into energy to use in the electron transport chain. However, you don't get this energy anymore, which is a major energy source for muscles. So naturally, these individuals get less ATP to their muscles, which means you have weaker muscles because the muscles use a lot of ATP. And so why are these more severe during fasting? Because under fasting conditions, remember, we have 2 sources of energy storage. We have fatty acids and we have glycogen. Glycogen lasts about a day. Fatty acids, depending on how many you have, can last you about, I think, like if you're really overweight, you can last like 15 months without eating off of fatty acid energy alone. The normal person has like 2 to 3 months worth of food just stored in themselves because of fatty acid. So if you're fasting and you run out of glycogen, the only energy you can get is through the fatty acid. But if you cannot deliver the fatty acid to your muscles, then your muscles are going to starve incredibly fast. So that's why it's important for these individuals to keep well fed. I think for these type of people, it would be a diet thing, because I don't think there's a supplement you can, like, that enzyme just doesn't work. So I don't think there's like a supplement that you can take to just make it work again. That enzyme, for whatever reason, is not sufficient. So my guess would be that you just have to have a diet that's low in fat, because you can't really utilize that for energy, and just more higher in carbohydrates as your energy source. That would be my thought on how somebody like that would manage that condition. All right. So moving on, let's get to the actual chemistry of what is happening when we break down fatty acids. To make your life easier, do I want to make your life easier? Yeah, I mean, we don't have to remember all this. If we just know, if we know what FADH2 goes, the electron transport chain, that's good enough for me. This is basically just going over like the electron transport chain and just words. So just know from FADH, that's where we're getting electrons to go into the electron transport chain. Same here, NADH, that's where we're getting energy. But let's see now how we break down fatty acids. And it's a stepwise process that is repeated many times on the same fatty acid. And get ourselves oriented. So here's our coenzyme A. The first hydrocarbon after our C double bond O here, we call that the alpha carbon. And then following that is the beta carbon. So the first step is to form a double bond between the alpha and beta carbon, and the beta carbon is going to be colored red for us. And this is done by acyl-coenzyme A dehydrogenase, or AD. Again, I mentioned this like 20 times already, but anytime you see the word dehydrogenase, you know you're doing a redox reaction. So you should expect an NAD or an FAD to appear. And in our result here is our trans delta 2 enol coenzyme A. Don't worry about like these specific names, the trans delta 2, the 3L hydroxy acetyl, beta keto acyl. Don't worry too much about those. Rather, if you know the structures, like what's happening structurally, and you know what enzymes are doing it, that's what I'm mainly interested in you knowing. So first, we have a dehydrogenase to make FADH2. That can go on the electron transport chain. So from the first step, we have energy already. Following this, once we have a double bond, we're going to use a hydratase. So a hydratase is kind of telling you what's happening. Again, we're going to add water to our beta carbon. So the beta carbon gets an OH. And this is carried out by enol coenzyme A hydratase, or EH. So double bond, OH. Following that, we have another dehydrogenase. And what's happening there is we're going to take the OH we just had, and we're going to form a C double bond O. And the last step is a thiolase. So a thiolase will cut a thiol bond. That's basically what's happening. But this enzyme's actually doing the reverse, at least in terms of the fatty acid, and that it's making a new thiol bond. But it is breaking the thiol that was in the coenzyme A. It's breaking that SH right here. That's what it's breaking. So we have a C double bond O on our beta carbon. Coenzyme A comes in, knocks off 2 carbons in the original coenzyme A. So you make Acyl-CoA, and we already saw this little guy, if it wanted to, could go to the citric acid cycle and be used up to make energy. While here, we have made a fatty acid coenzyme A again. The only difference now is that it's 2 carbons shorter. And so we just repeat the process. And the process is, we're always focusing on the alpha beta carbons, double bond OH, C double bond O, cut. Double bond, OH, C double bond, C double bond O, cut, right? When trying to learn this, always keep in mind, we are trying to remake the compound at the very beginning of this process. We are always trying to make fatty acyl coenzyme A. That is the process here. So we are removing 2 carbons at a time and doing 1 round, but we always end with fatty acyl coenzyme A. And the atoms that really matter here are alpha and beta. So we form a double bond between the alpha and beta. We add an OH on the beta, C double bond O to the beta, then cut away the bond between the alpha and beta and replace it with a coenzyme A. And that is beta oxidation. So any questions about that process now that you've seen it worked out? Does the 2 fewer carbon come from that cleavage? Yes, because when you cleave at the very last step, what comes off are these carbons. This CH3 used to be the alpha carbon. This C double bond O is this. So everything in black is what we're losing. And the red molecule is, we're going to just start the process again and again, lose 2 carbons. So every time you do beta oxidation, you lose 2 carbons from your fatty acid. But when you do this, you gain an FADH, you gain an NADH, and an acetyl coenzyme A, which, as I said, can go to citric acid cycles. So you can see how these fatty acids have so much energy compared to like a glucose molecule. You can get a lot more energy per gram of fatty acid versus per gram of glucose. Now the first step here, acyl-CoA enzyme A dehydrogenase. These only work on specific fatty acids. So we actually have 4 different versions of this enzyme, and they're just for different tails. So we have an acyl coenzyme A for fatty acids that are 4 to 6 carbons long, one that's for 6 to 10 carbons long, and then you have one that are between like medium and very long. And then you have one for very long, which is 12 to 18. So different acetyl-CoA enzyme A dehydrogenase will come in and work on different fatty acids for us. And that's just because, you know, when you bind to this molecule, you have to accommodate the fatty acid tail. So different versions of the enzyme will accommodate different tail lengths here. All right, so any other questions before I move on? All right, now I have a worksheet, and this worksheet is basically like 4 or 5 questions that deal with beta oxidation. So I'll put that up online. Instead of just having like question in a row here in class, maybe I'm just going to put that up, and I'll put the answers up as well at a little later date. But that's a way to study for this. So just be on the lookout for that in the next day or 2. I'm going to put it in the test 3 folder, beta oxidation worksheet. All right, so let's talk about some problems with beta oxidation. And there's basically 2 problems that you can run into. So problem 1 is fatty acids that have a double bond. also known as unsaturated fatty acids. Because if we go back to our process, right, our first step is making a double bond between the alpha and beta carbon. If there's already double bonds in the structure of our fatty acid, this can cause problems. So that's what we're going to look at first. But just to give you an idea of things to come, our second problem in breaking down fatty acids is if you have an odd number of fatty acids. So when we are doing beta oxidation, we always break down the fatty acids in units of 2. However, if you have an odd number, this can become a problem. So we're going to look at problem 2 in a few slides, but we're going to look at problem 1 right now. What happens when you have unsaturated fatty acids? So let's look at linoleic acid here. Linoleic acid has a double bond on carbon 9 and carbon 12. And so you can do 2 rounds of beta oxidation, no problem. So we lose 4 carbons. So 4 carbons gone. So now we have a double bond on carbon 5 and carbon 8, which was just 9 minus 4 and 12 minus 4. And when you do the next round of beta oxidation and you do your first step of acid coenzyme A hydrogenase, if we do our numbering scheme, so this is carbon 1, 2, and 3. Carbon 2 is the alpha carbon, carbon 3 is the beta carbon, right? So we put a double bond between 2 and 3. Now we have a problem. And the problem is that our double bonds are becoming much too close to each other. And our enzymes are going to fail to work here when we continue the beta oxidation, right? So we're going to look at the left-hand path here. So let's say that you do beta oxidation with this double bond here. So you do beta oxidation, you lose 2 more carbons. So you lose these carbons right here, they go away, and you're left with this molecule. Before you do anything in your next step of beta oxidation, you have what's called a beta gamma double bond. So this is the alpha carbon, that's the beta carbon, that's the gamma carbon. And this double bond here is not what we want. Remember, we want the double bond between the alpha and beta only. We cannot do that currently because the beta is already in a double bond. But that's really not that big of an issue for us because we have an enzyme called enol coenzyme A isomerase. What this isomerase will do is that it'll take this beta gamma double bond and move it over. So now it's an alpha beta double bond. So instead of a dehydrogenase, we have an isomerase to take care of this. What that means is we don't get electrons to FADH2. So we're losing out on energy by having this double bond here. But by doing the isomerization reaction, we can at least continue the breakdown of this fatty acid, which is what we do, right? So we can now continue beta oxidation. So we do 1 round of beta oxidation and we lose 2 more carbons. So we lose another acetyl coenzyme A. So this goes away, this goes away. And what we're left is, if we do 1 more round, so we get rid of our Acyl-CoA enzyme A here, and we do the first round of beta oxidation, where we have our alpha and beta double bond here. But this leads us to another problem in this fatty acid. Delta 4 double bond. So if you remember nomenclature back from biochemistry 1, where we talked about fatty acids, delta 4 is simply just saying on the 4th carbon, you have a double bond. Again, this will cause a problem because these double bonds are connected. And we don't want any double bonds near our alpha and beta carbon. The enzymes can't work with that. So to get rid of this, to get rid of the delta 4 double bond, we have to use a reductase. So this path is for mammals. Over here on the left, that's for E. coli. We're not going to really talk about E. coli, so we're just going to focus on the mammal version of this enzyme. So here in the mammal version, we have to burn an NADPH, not an NADH, but an NADPH to NADP. And we take these 2 double bonds, and we're just going to make a 3,4 double bond. But this is easy enough to fix. We have another isomerase, this time a 3,2 isomerase. And we can continue beta oxidation because we move this double bond from the 3,4 position to the 3,2 position. Or, I know this is confusing because I'm using both numbers and letters. Yeah. Not my idea to name fatty acids like this, alpha, beta, gamma. We move it from the beta gamma double bond back to the alpha beta double bond, which is what we want. And then beta oxidation, there's no more double bonds to deal with. And so you can continue moving or chopping up your fatty acid in 2 carbon chunks. So that's one problem with double bonds, or 2 problems combined. But something else can happen with your double bonds. So 3,2-enol coenzyme A isomerase. This enzyme is the same as this enzyme. Same exact enzyme. And let me just rewrite what it does. What this enzyme does, it takes a 3,4 double bond and converts it to a 3 double bond. So it's just moving the double bonds between different carbons. But this is reversible. This enzyme can go either way. It can take a 2,3 and make it a 3,4. And so I find it's always important to point this out. Enzymes don't think, right? They don't know that taking a 2,3 and making it into a 3,4 is quote unquote bad, because we can't do beta oxidation if you have a 3,4 double bond. It needs to be a 2,3 double bond. But this enzyme works both ways. It can take a 2,3 and make it into a 3,4. And that's what our problem 3 here. If we have a 2,3 double bond already, before we've done anything with beta oxidation, what can happen is this 3,2-enol coenzyme A isomerase will flip that double bond from a 3,2 to a 3,4, which just breaks beta oxidation. Beta oxidation no longer works because the double bond's in the wrong place. And also, you're starting to get conjugated double bonds here, which is bad. You don't want conjugated double bonds when you're breaking down fatty acids. The only place you want a double bond is at the alpha beta position. You don't want any double bonds near it. So we need to fix this problem. If you get a 3,5, the 8 really doesn't matter. It can be anything past here. The only thing that matters here is a 3,5, and that's why it's highlighted in red. If you get a delta 3,5 fatty acid, we need to fix that using an isomerase, which is called by the lovely name 3,5,2,4-dienyl coenzyme A isomerase. But what this isomerase does, it changes a delta 3,5 into a delta 2,4. So it's just moving the double bonds over one position. Now we are in a position where we've seen this already, because over here we have a delta 2,4 as well. So now it's the same thing, where you use your reductase, change your delta 2,4 into a delta 3, then use an isomerase to change it to a delta 2. This is done by isomerase. This is a reductase. This is also an isomerase. Then this will run, but we have another double bond over here at the 8 position. And so eventually you're going to run into the delta 2,4 problem again. So just to reiterate, what are the problems here? So this first problem is if you have a beta gamma double bond, right? So if you have a beta gamma double bond, you need an isomerase. So beta gamma, you move it to an alpha beta through an isomerase. If you have a 2,4, or I guess another way to put that is alpha and gamma. But if you have a 2,4, you use a reductase, and that's shown over here to make it just a delta 3, and isomerase to make it a delta 2. And the other problem is if you have a 3,4 on accident, then you can just do the isomerase, reductase, isomerase. So that just deals with beta oxidation of fatty acids that have double bonds in them, unsaturated fatty acids. So a lot of information on just this one little slide. So I'm sure this is one of those slides that you need to sit down with and try to digest of what's going on and working out the logic. But if there are any questions, just to begin with over this preliminary first explanation of what's going on here. Now I know it's confusing, very confusing about what's going on, but there is some logic to it. So it's all about moving and softness, right? Anything that would prevent beta oxidation completion. No, I don't think there is. I don't think there will be a fatty acid that would stop beta oxidation completion. Yeah, because we have enzymes in our body that can fix problems with double bonds like this. So that should be fine. These are just the common problems that come up when you eat an unsaturated fatty acid. So we have enzymes in our body that can take care of that. Okay. So let's look at the one issue, though, of fatty acids. So the caveat to that last answer is if you have an odd number, then yes, at the very end of that odd number, we cannot continue beta oxidation. So if you ever end up with an odd number fatty acid, eventually after beta oxidation, you're going to end up with propanol coenzyme A. So if you remember your organic chemistry, prop is the prefix for a 3-carbon compound. So propane is CCC, right? A little blast from the past from organic. So propanol coenzyme A is 3 carbons with coenzyme A. And since we remove 2 carbons at a time, we cannot do beta oxidation properly with something that is just 3 carbons long. So what we do is instead of creating acetyl coenzyme A to go into the citric acid cycle, we're going to make succinyl coenzyme A and put that into the citric acid cycle itself. And this is done in 3 simple steps. The first step is we're going to use ATP and attach carbon dioxide to this propanol coenzyme A. So we're going to take this 3-carbon compound and make it a 4-carbon compound via carbon dioxide, and this is done by a carboxylase. Usually when we look at carboxylases, they are removing CO2. Here we are adding CO2, and this is an unfavorable reaction, so we need to add ATP. And this will make methylmalonyl coenzyme A. And then after that, we have to switch chirality. So in our original reaction, we make S, but we need to switch it to R. So this is done by a race mace. So you probably talked about this in organic chemistry, where when one of the big problems of synthesizing things is that you can end up with an equal number of R and S molecules, which isn't really good depending on your plans with the molecule, depending on what you want to do with the molecule. And here in biochemistry, we've never really run into this problem all that much, where we have a molecule of one chirality and we need to make it the other one. So this is the first time I believe we've ever talked about an enzyme with this type of name. So this type of enzyme will simply switch chirality. So we're switching it from an S to an R. If you don't remember how to count like what's an S and what's an R, like how to look at a carbon and decide that, don't worry, I'm not going to ask you that on a test. I'm never going to give you a molecule and say, is this S or R? That was organic chemistry, and we're not too worried about this. But we just need to go from S to R here. And next is a mutase. So this whole time, we've had our carbon dioxide here attached to the carbon that is right next to our C double bond O. So CO2 was added right here. However, to make it to succinate, or rather succinyl-CoA, we take our carbon dioxide and move it to the very end to make succinyl coenzyme A. This is done by a mutase. Mutase just moves around our functional groups here. Succinyl coenzyme A, hopefully you know that is part of the citric acid cycle. Can be fed into there. And that's how you deal with odd number of fatty acids. So you break it down via beta oxidation until you get 3 carbons left. And then instead of doing more beta oxidation, you just add a CO2 to it and make succinyl coenzyme A. So that solving odd number fatty acids is a lot simpler than unsaturated fatty acids. But any questions about that process? All right. Now, for Succinyl-CoA enzyme A, we actually need to tweak it a little bit before it can be put into the citric acid cycle. So from fatty acids, we don't actually take that succinyl-CoA enzyme A and just shove it and make it into succinate. You just have to convert it to pyruvate first. And then pyruvate can use the pyruvate dehydrogenase complex. And I hope you have studied the PDH mechanism for Thursday, just a hint. I'm not going to ask you to name the mechanism like step by step, but I will ask you about the mechanism itself and about PDH itself. So make sure you go and review that if you haven't yet. There's a couple questions about that. So we need to take pyruvate and then we change it into acetyl coenzyme A to enter the citric acid cycle. Conversely, to do this, we need to convert succinyl coenzyme A to malate. Remember, malate is one of those molecules that can be transported across the mitochondrial membrane. Again, if you don't remember those transport systems, just review yourself on those as well by Thursday. Hint, hint, nudge, nudge. So we take our succinyl coenzyme A, make it to malate. Then we can transport this malate to the cytosol, do some decarboxylation, make it into pyruvate. And this is done by, not by malic enzyme. Then we take that pyruvate dehydrogenase citric acid cycle. So just some steps there in the middle before we can take the succinyl coenzyme A and shove it right into the citric acid cycle, make it into malate to pyruvate to acetyl coenzyme A. Making it into malate is simply for shuttling reasons, so it can move across the membrane. And then pyruvate using pyruvate dehydrogenase. Again, make sure you review pyruvate dehydrogenase. Let's see. And our last slide here has to do with peroxisomes. Now, to break down fatty acids, that usually happens in your mitochondria. That's where that's going to happen. And the citric acid cycle, again, happens in your mitochondria. So it's kind of convenient because we're making acetyl coenzyme A that can go directly to the enzymes it needs to go. However, mammals also have peroxisomes. Peroxisomes are used to break down really long fatty acids. So if you eat fatty acids that are extremely long chain or have branches, the peroxisomes are going to be the ones that take care of that. And it's going to shorten them until they can be broken down by the enzymes in your mitochondria. So here we just need 3 different enzymes. And I'm going to say, don't worry about like the names of these. Just know what peroxisomes, what's the point of peroxisomes for beta oxidation. They take care of long fatty acids and shorten them. That's what we really need to know for that. Just make the decision of just this top portion. And that is it for the information I had to cover for today. So there is 10 minutes. Anything you want to know about test 2 in these 10 minutes? Anything at all? Or anything that we covered today? Any questions? So the test material is 16.4, 18.4 with no lipid material. I believe that's correct. No chapter 20, for sure. Chapter 17, yes. Chapter 18, yes. Let me just the tail end of 16, I believe. Let me just double check my spreadsheet. Oops. Yeah, mainly focus on 17 and 18 for the most part. That's where the bulk of the questions come in. Yeah, gluconeogenesis. So that's the only part of chapter 16 that's going to be on there. Then all the citric acid cycle regulation, the different uses of the citric acid cycle, of course, the actual citric acid cycle, and then the electron transport chain where we looked at the different complexes, we looked at ATP synthase, we looked at regulation, that's it. Nothing to do with lipids, nothing to do with glycolysis. So the length of the test, I'm looking for roughly 40 questions. Of those 40 questions, maybe 8 will be short answer. And all of those short answer, like I would say like 4 of those can be answered in a sentence. And the other 4, maybe like 3 sentences max. So it is really short answer. So if you know, and some of the multiple choice will take a little bit of thinking because it's like, here are 4 options. Tell me which one or multiple ones are true. So some of them will take you some thinking about. Others are just straight like, do you recognize it? So I think if you know the material, you should have no problem finishing in time. The points, I haven't like made the points yet. So if you are looking at like, I don't know if I made it visible to you or not, but like you might notice a column called test 2 in your grade book where it has like 400 points or something like that. You can pretty much ignore that. The points I'll probably decide on Wednesday night, like what each question's worth, but that's the layout of the questions. And I'll even say a bonus for sticking around with me all day today. I'm not going to ask you to do a calculation, so you can safely ignore that. There is a calculation we went over in this section in the electron transport chain where we were calculating delta E through different redox centers. I already asked that in test 1, so I'm not going to ask that again. So don't worry about that. Also, I mean, the hint from that came from I never put up an equation sheet either. So the IMS and matrix. What about the IMS and matrix? That's so the intermembrane space in the matrix is where the proton gradient is created. And your electron transport chain is going to be in the membrane between those two. Oh, that was one of the math examples. Yeah, pumping protons. Anything else? ATP synthesis mechanism. sure. We can look at that real quick. Let me pull up the correct slide. Is that 311? Let's control. Okay, there it is. Let me share. All right, so the way that ATP is made in ATP synthase is that you have alpha beta subunits in F1. You have 3 copies of each, 3 copies of A or alpha and 3 copies of beta. 1 alpha and 1 beta form a complete unit. And these alpha and beta subunits change conformation through the rotation of F0 that we talked about. You know, the protons are going through F0 like a carousel, and this is what's helping to change conformation. The alpha beta subunits themselves don't move, they don't rotate, but they change conformation. And you have 3 different, oh, I should turn on my camera so you can actually see me saying 3 different conformations you can be in. The L, the T, the O. The loose, the tight, the open. All right. So if we start on the bottom left here, we have open, loose, and tight. And so our first step is ADP and phosphate, they bind loose, right? They will bind, that binds to the loose. And then energy comes in. And the way that I think about this is that if you're loose, you go to tight because you have your ADP and your phosphate in the loose conformation. And then in tight, you're like shoving them together to make them to ATP. So it's like more open, now it's closed. You're putting that phosphate with the ADP to make ATP. What was tight now opens up. Because if you were tight, that means you made ATP. And so you want to open up and release that ATP. If you're open, you're going to become loose because now you're ready to get ADP and phosphate again. So if we just focus on one alpha and beta pair, and maybe it'll help if I don't have the share here. Do this with my hands. So we start in the open conformation. Nothing is there. Let's see, what do I have around my desk? We go from open to loose, where we have ADP, my pen, and this lighter will be phosphate. So that's loose. We go to tight, smash these 2 together to make ATP. And then we go open again, and they fly off as ATP. So open, nothing there. Loose, bind ADP, phosphate. Tight, ATP, open, release. That's the mechanism in a nutshell of the F1. There, remember, there's other parts to go with that where you have the hydrogens in F0 spinning around. And that is what's causing this conformational change, but that's it in a nutshell. And with that, that was the end of class. So if you have any other questions, let me know. I'll be happy to answer them. Otherwise, look for that link to appear starting at 12 a.m. Thursday, and it'll close at 11:59 p.m. Thursday. So you have all day to take it whenever you want. You have an hour and 15 minutes. Otherwise, good luck and see you next week. Have a good one.